But we have a great program tonight on the topic of commercialisation of biological science with two wonderful speakers, Sally Gras from the University of Melbourne and Robert Klupacs from the Bionics Institute. Clearly a topic of current interest, particularly noting the recent announcements from the Victorian government regarding the need for an mRNA vaccine facility and more broadly for the focus of the federal government on modern manufacturing. Commercialisation of biological systems is essential to Australia's health and prosperity, but really requires a truly interdisciplinary team of engineers and technologists working alongside biologists, and I think you can see some of that in the presentations tonight. So our first speaker tonight is Professor Sally Gras, well known to me, but who is Director of the ATSE Dairy Innovation Hub at the University of Melbourne. She's also an Associate Director of Bio21, Molecular Science and Biotechnology Institute, and leader of the Food and Agribusiness Research Team within the University's Faculty of Engineering and Information Technology. Sally has a unique set of cross-disciplinary skills, having trained as both a chemical engineer and a molecular biologist. She received her PhD in protein biophysics from Cambridge in the United Kingdom. So I'll hand over to Sally to give her presentation.
So you already saw in Sandra's introduction that I have a background in biology and chemical engineering, and I've got my feet in both the engineering and biology camps today in terms of where my laboratory is based and the teaching I do as well. And my particular research interest is in food and biopharmaceutical manufacture, and I've worked with a number of companies shown up here over both industries over the last few years. And today, as Sandra mentioned, I'm going to talk about commercialisation of biological science, which I'm going to take in the broadest terms, and I'm including food amongst that as well. And I'm also going to talk about innovation and translation in these industries. So why food and biopharmaceuticals to start off with? Well, the UN's identified that we estimate more than 2 billion more people will need to be fed sustainably by the year 2050, and that's a significant challenge. And the COVID pandemic's also shown us the need for local manufacture as well. And so front of mind, of course, is the vaccine production, but actually up to 90% of our medicines are imported. So there's a sovereign need to make our own medicines. So it's important for Australia's health and wellbeing, but also for the economy. So collectively, these activities are worth around $12 billion to the economy each year. So the key question then is how do we use technology to assist industry to address these challenges and also to achieve sustainability goals? And when it comes to technology uptake and commercialisation, I particularly like, and you can't see it up here because of so what it says is 10 simple rules to commercialise science. And of these, my favourite is that there is no single path to commercialisation. And so you can see I've put there 4 different routes to commercialisation, spinning out companies, patenting, licensing, and I've also put translation through contracts. And what I mean there is where you build the research capability, perhaps in a program, and then you apply that through a commercial contract alongside the research. And the top 2 of these get a lot of attention. And recently the Minister for Education talked about our performance on these top 2 metrics. So startups per billion in research and invention disclosures. And you can see that Australia doesn't perform as well as its international competitors here. But I'd like us also to think about the other 2 on the list here. So licensing and translation through contracts. And from the research that I've led and also the research that I've been involved in, the bulk of the translation has occurred along here. So through contracts, through licensing and some patents. So I think that we can miss something if we forget about these activities when we consider commercialisation. So I'll show you some examples of the types of activities we've done both in food and biopharma through these types of routes, and talk a little bit about the benefits and the challenges that I've identified as well. So over the last 7 years, I've led the ARC Dairy Innovation Hub, which was a $13 million program with Australian Research Council funding, and also funding from the Dairy Innovation Hub, from DIAL, and 5 of Australia's most prominent dairy manufacturing companies. We had a chair in Fial, the Growth Centre, and we also had 2 universities involved, so the University of Melbourne and the University of Queensland. And this was quite a successful venture, and I'll show you some examples of that. But one of the great successes coming out of this program was that we then led into a second program, called the Dairy Innovation Hub. This is a 3-year industry translation program, so with direct industry funding, 3.8 million, and 4 of Australia's most prominent dairy manufacturing companies. You might notice a difference here, and that's because there's been quite a lot of shifting in the dairy manufacturing industry. So one of the key focuses of this second program, which is very much industry-based, is around translation as well. And that's through a mixture of pre-competitive and competitive projects. But the main mechanism is through licensing and then these translational contracts that I'm talking about. So to give you a sense of, so what I wanted to share with you is about the first hub that I led and some of its impact and the translation that we've done with industry. So I mentioned already that there was a $13 million investment, and we sat down with industry and we looked at how have we contributed to either increase sales or benefits in those businesses, and we identified over $250 million worth of benefits. And around 200 of those were realised already, and around 50 that are hopefully going to be realised in the future. In terms of our more recent hub, it's still fairly new. It's only been around for a year. And so we don't have any impact figures like this just yet, but I can say that we've got really effective engagement with industry. And I think this is really important for translation. It's the first step. And so over the last year, we've had 120 technical meetings with 36 industry representatives. So you can see we've been busy. So I'd like to then show you just how we've translated some of the techniques and capabilities that we've developed as well. So within the hub, we had 5 research themes, which are shown here. And what we tried to do is to establish capabilities, so the ability to do different techniques and build fundamental understanding both within the university and in industry. And I'll talk, a number of these things also looked at technology evaluation, which I know is also important to the academy. I'm going to talk specifically about the microstructures thing here, which I led, and how it looks at food structure at a micron scale. So what this is underneath that heading there is our biological toolkit. And so what we use as the micron name suggests is a whole lot of microscopy. And this is based at the Bio21 Institute, so it's the biological bent to this type of research. And we combine this with a whole lot of analytical techniques and things that are available on the Bio21 platforms. And by combining this with process engineering, we can really help manufacturers. And our favourite types of microscopy that I'm going to show you today are the confocal microscopy, which gives you these red and green images where the proteins in green and the fats in red, and cryo-SEM, which gives you these grayscale images. And these are fat droplets you can see here, and the scale bar here is 10 microns. So how we've applied these types of biological skills together with process engineering to help manufacturers. The first of these examples is around benchmarking processes. So cream cheese, for instance, if we're interested in this type of product, we might ask, how does the structure develop in a product? And by taking all the various steps involved in the manufacture, you can see here from milk on the left all the way through to cream cheese, we can build up an understanding. And from doing this, we can help manufacturers see the importance of particular operations. So it might be separation, or it might be heating and heat exchange. And we can look at the impact of process variables, formulation or shear, and look at that both in the lab and at scale. And working across scales is really critical, I think, to translation as well. And we do a lot of scale up, of course, but we also do scale down too. So taking processes and doing it high throughput at smaller scale in the laboratory. Another type of activity that we do is benchmarking a product category. And so this is back to cream cheese again. And if you were to take a selection of products from your supermarket shelf, this is what they look like under the microscope. And that's actually very valuable for manufacturers because they're able to benchmark a particular product against the whole field and understand where they sit and why their product may differ. It's also quite important for Australian manufacturers to be able to increase exports, particularly into Asia, for example. And to do that, they need to be able to increase their manufacturing capacity, which sounds simple, but it's not necessarily so. And we also know that manufacturers want to avoid capital cost in these exercises and also de-risk or reduce the risks around expansion. So we've worked with manufacturers, and this first example here was we saved them capital costs because we helped them understand the product structure and the effect of brining. And so reducing brining time and pressure, so these types of products you see here, enable these types of savings. Another thing that can happen when you increase manufacturing is you change your product. So you may not fully understand all aspects of the manufacturing process. And so you can then end up with brand damage where you change consumer perceptions or customers in general. So we've helped companies to avoid this by increasing their product and process understanding and ultimately help them achieve an increase in capacity. And I think most rewarding in these examples was that eventually this became part of the design specifications for expansion, the type of work that we were doing. Another example is looking at the product. So microscopy, again, lets you be those eyes at a different length scale. And this is an infant formula product, and you can just see under here, these are big air vacuoles inside a little powder particle. Or you might be looking for free fat that you see here. And so we've helped companies to assess and troubleshoot those defects. And in one case, we helped to resolve production problems contributing to that change, which then led to an increase in 8 million of sales in the first year. Another area we've assisted is in product export. And again, if you look at cream cheese, this type of product and similar products in this category, when they are exported into different countries such as Japan, they are then assessed by a range of standards associated with customs. And so by providing independent verification and looking at quality prior to shipment, we can reduce risk, and again, that can help manufacturers, which adds up over a consignment. So hopefully what I've shown you with the food example and applying biological skills in that case, I've shown you some of the benefits of translation. So I think it lets you apply a fundamental skill set to achieve commercial benefits. I think one of the great benefits is, of course, repeat business for everyone, and that's also the companies. And I think it lets you look at identify and then work on problems that are important for industry. It also helps to achieve impact, and here's Lydia Ong, who's within my group, being recognised with an ATSE award for some of this, and it helps us work on relevant samples and at relevant scale. And one of the things we find is that if you look in the literature, for instance, looking at the freezing and thawing of dairy products, we find that people are working on things at this type of scale, a little test tube. And as you can imagine, the heat transfer in a pallet is completely different. So you have to work at scale on something that's relevant to industry to understand fully what's going on. And I also think that the site work is really valuable for staff and for students, and having an extended team and greater expertise, a two-way learning process, and ultimately employment are all benefits. So the second example I'd like to give you is more of a traditional bio example, and that's biopharmaceutical manufacture. And we do a lot of work with different types of molecules, but I'm going to focus here on small molecule manufacture. And you may be interested to learn that Australia is actually a world leader when it comes to supplying opiate starting materials for opiate drugs. And these drugs are grown in the form of poppies in Tasmania, and they're extracted both in Tasmania and in Victoria. And the companies involved in this type of processing are shown over here on the right. And this is quite a different area. When it comes to commercialisation and translation, it's quite different to the food industry in that there are a lot of patents. But equally, there's also confidential know-how, and there's still the licensing, and there's still translation through contracts that's occurring too. So there's been a lot of science in this space, which is very exciting, and there's been an increase in the yields that you see as a result of this science too. So the morphine and codeine that are produced in these poppies This has been increased by science-based approaches. These are used to form pain drugs, and the vein and oroparvin are intermediates that are then used to manufacture either drugs for treating pain or anti-abuse drugs. And it's because of this science that the yields have increased from 3 to 40 kilograms per hectare. And an example I'll draw upon is the Molecular Biology and Breeding Program, which was initially funded by GSK, which was then sold, the division was sold to Sun Pharma. And that was complemented, or I should say as well, that resulted in a whole lot of science papers that you see here, predominantly led by Ian Graham out of York. And this was matched by and complemented by 20 years or so advances in solvent extraction that Jeff Stevens led here at the University of Melbourne in chemical engineering. But with all of these advances, there are still challenges in the industry. And one of the key challenges is that it takes a long time. So the lead time to go from planting a poppy all the way through to the drug is at least a year or so. You're planting in winter, you're harvesting in autumn, and by the time you get here to your final drug product, it's a long time, and you can't anticipate necessarily what you're going to need here in terms of the market. So that creates a big challenge for the industry. The alkaloid yields also vary, and so the whole approach is really quite inflexible, despite having good yields. So a new approach that many people are working on worldwide is about using synthetic biology in engineering. And I guess one extreme end of this approach is the synbio or wholesale engineering approach, where you just take a yeast cell, or it could be a bacterial cell, a single cell, and you take very simple feedstocks, so maybe it's glucose, and you're putting it contained in a laboratory and converting those simple feedstocks through a series of steps, which are very small, but you can get the sense that this is occurring within the cell. And that can then produce a drug in the laboratory. So this example here is scopolamine. This is from Anthea, one of the leading companies in this space, led out of Stanford by Christine Smolk. And this approach would really radically change the whole industry. So it's quite exciting in that sense, but also quite a commercial challenge, although Anthea have indicated that they're now at pilot scale. We've taken a slightly different approach. You might consider it a lower hanging fruit approach, and we're taking a hybrid of pairing select enzymatic reactions with existing technology. So the advantage of doing this is that you can then interconvert between your stockpiles to balance market demand. You can make use of all of that science that's already occurred, that's given you the great yields, and the existing infrastructure. So it's potentially faster commercial uptake as well. So I'd just like to give you one example of what we've been doing in this space, which is around codeine. And so codeine is a World Health Organization essential medicine. But the way it's currently made is that the morphine is extracted from the poppy, and then there's a synthetic process that's not terribly sustainable. And the approaches to make codeine-rich poppies have been successful, but they still have some risks associated with them. So you can't see it again underneath this, but you just have to take my word for it, that you've got the vein and it's an intermediate, it's all right, I've got other stuff that will come up in a second. Here we go, I'll show you here. So we've got the vein as an intermediate feedstock, and we're using bacteria with 3 key enzymes, and we're converting it through to codeine directly. And what's interesting in this type of space with symbioology is there's lots of really exciting, high-impact proof of concept papers that are great to read, but we've noticed that a lot of them are, again, produced at this scale, and the yields are very low. So commercially, there's a long road still to get there. So we've been working with Sun Pharma and QUT, so Rob Spain at QUT, to try and address some of these challenges. So in this work, by looking at how we spatially and temporally move the reactions about, we've been able to increase the conversion of feedstock from 2 to 64% and have a 160-fold increase in volumetric productivity, which makes it similar to other commercial products in terms of productivities. So we've been able to take something from proof of concept to something that's more likely to be adoptable by industry. And I think working on these types of challenges can then assist uptake by industry. And interestingly, this work was published in 2020, And it came shortly after the US government reduced opiate quotas. So they were redirecting material from oxycodone and hydroxycodone. And those same feedstock stocks, of course, were going through this process to then make a WHO list of essential medicine. So redirecting to a better use. But there are challenges in the commercialisation of these types of areas, and the first of these is around multidisciplinary training. So if you're taking a standard synthetic process chemistry approach, you really need to train as well in biology if you're going to try and introduce some of these approaches. And conversely, if you're taking a syn biology, there's a fermentation approach, you need to have the engineering to understand the downstream processing. And without that, it's not going to work. The equipment, there are challenges around conversion of chemical infrastructure. And also internationally, there's really not many facilities for this type of downstream processing of large-scale fermentation of pharmaceuticals. An area or an example of where we've been working in these areas is with Riverstone Biotech Australia, a small biotech company, and we're making, with them, we're helping them to make drugs that treat addiction in a more sustainable approach via fermentation. And these are the types of engineering challenges that we face around impurities and dilute solutions. So very typical engineering challenges that we'd like to address to enable commercialisation. So hopefully, in summary, what I've shown you today is that there are many routes to commercialisation, or at least I believe there are, and that licensing and translational contracts, I believe, are a lower risk routes to uptake and to impact within companies. I've shown you some examples of benefits and how they can occur to both sides, and how we can combine biology and chemical engineering. And yes, it can introduce challenges around training and equipment, but it also provides a whole lot of opportunity for impact as well. And finally, I'd like to say that I think that university research can really make a tangible difference, from the types of simple translations I've talked about to paradigm shifts, but the industry is critical to that translation too. We've been very fortunate to have very engaged industry partners who are involved also in training and ultimately co-investment as well with the government, all by themselves. And I think it's a really exciting time to be working with Australian manufacturing. We think about a manufacturing-led recovery from COVID, and also how there's a lot of emphasis at the moment on increasing the reach and impact of research. And this is an area that's really ripe for impact through translation and commercialisation. So I'd just like to thank all the people involved in the research that I've been talking about today. So for each one of these people, there's probably 2 to 3 industry people too. Much harder to get their photographs, but we also really appreciate their input too. We have a range of collaborators that are shown here, and also funders. And finally, thank you for your attention, both here and online.
Thank you, Sally. And we might pause there to see if there's any questions from the audience. If you are on Zoom, can I also just let you know that you can lodge a question through the chat, and Mark is happily taking those questions. So I'll first throw to the audience, though, do we have any questions from the audience for Sally? Doreen.
Thanks for your great talk, Sally. I was quite interested in your mentioning that the industry or the cheese manufacturers were able to compare their products from your examples, your microscopy samples that you showed us. So were they agreeable with comparing their product with each other's, or how did that all work?
Sometimes it's tricky.
Yeah,
So often what we would do is to benchmark a whole product category, and we would de-identify the range of products, and then we might say, but your products this one amongst them. But equally, sometimes you can work backwards looking at protein and fat composition and probably know what competitors want. So, yeah,
really interesting. Great, good to hear.
Mark, do we have any come through?
No.
Any other questions from the audience? Mark.
Sally, you've obviously you're doing a lot to improve industrial processes. Can you tell us roughly what percentage of your funding comes from industry? But clearly, you might help companies to be more profitable and be more efficient.
I'm not sure if I can give you an exact percentage, but perhaps I can clarify that certainly the hub, the ARC Dairy Innovation Hub, was very much focused on translation, and that had, as I said, 13 million. I think it was 5 million of ARC, so we had a significant portion of industry funding. And then the subsequent hub that is now this translational focus is all direct industry funding. But other work that I talked about has been sponsored by linkage grants and a range of different routes. So I guess we've taken traditional research routes through match funding, but we also use state government funding too. So we've used things like the Innovations Connection Scheme and the voucher scheme, and these can be really valuable for smaller companies and enable you to do smaller projects too. So I guess a lot of our funding, but not exclusively, involves both federal government and industry, and some is industry owned.
If there's no questions, I might take the chance to ask a question for myself. I'm interested in you describing where the technology is going in that, the pharmaceutical industry, for instance, is predominantly a batch process at the moment. Where is the industry moving in terms of Industry 4.0 and generally upskilling technology?
There's lots of aspects to that, but I guess, in terms of, you're right in that a lot of pharmaceutical processes are batch because of, or one of the reasons being that there's a lot of compliance and regulation and systems set up around those batch processes. And I think the industry recognises that there could be a lot of economies to eventually move towards continuous processing. I think that's happening around the world, but there are a few examples that are continuous and a lot of interest locally as well on continuous processing. So it will be interesting to see that get adopted, I think, both within small molecule and larger molecule manufacture. In terms of Industry 4.0, that's a really big area that has many different parts to it. I think one of the things that we've looked at with industry recently is the need for digitization. We know that there's lots of really big process data sets that are being generated in pharmaceutical manufacture, and I think there's the potential for these to be better utilised. So I think that's where there's going to be some advances hopefully in the future as well.
Thank you, Sally. And that's probably right on time. So if we could give Sally a round of applause for a very round up if you want to ask more questions. Let me introduce our second speaker, who's Robert Klupacs. Robert is Chief Executive Officer of the Bionics Institute. He has over 30 years of international corporate experience in technology development, mainly in the fields of medical technologies and biotechnology, with a particular focus on healthcare. Robert is scientifically trained in pharmacology and biochemistry, but he's also an Australian registered patent attorney. So again, I think there's a strong theme here of that cross-disciplinary expertise. And Robert has unique skills in translating and commercialising early-stage intellectual property into products because of that expertise. So let me welcome Robert to the stage.
Thank you, everyone. It's a pleasure to be here, and thanks for pushing back your dinner by a little bit to accommodate me. I really appreciate it. Just before I start, I have to say it's an incredible honour for me to be here. To be sitting and being a member and a fellow of ATSE is a bit intimidating, and so COVID didn't give me a chance to meet you all last year. It's my first outing, so thank you very much. Before I move into the presentation per se, Mark asked me when he rang me and said, look, can you give me some background about yourself, about why you should talk about it and what you're going to say is relevant to innovation? As you heard, I'm a patent attorney. I've been involved in this for 35 years. You mentioned biotech, but I've moved from biotech, and within biotech, you've heard there's always bits and pieces that Sally just spoke about. I've been involved in antibodies. I've been involved in small molecules. I've been involved in diagnostics. I've been involved in gene therapy. Seen a lot of that. I've also been involved in ag tech. I'm chairman of an ag tech company. And now for the last 7 years, I've been involved in medical devices. innovation at all levels is different, but the issues are all the same. I'm going to use medical devices primarily as my thesis tonight, but I believe, as I said before, it overlaps with everything that we do in innovation. I have to say up front that I saw my chairman of my institute earlier before, and I told him I was doing this talk, and he said, "That's fine, Robert. Tell them your opinion. Just remind them it's not the opinion of the Bionics Institute." So I'm putting that on the record to everyone. My hope actually is I can give you, and it is my opinion. I've been trained by some of the very best, and I'd like to acknowledge that the person who probably pushed me down this road taught me a hell of a lot. And some of the things he taught me some years ago I disagreed with, I probably agree with now, and that's John Gras, AO, sitting there. He put me on this journey. Probably Australia hates you for it, John, but I thank you for it. I think tonight I'm going to give you a slightly different perspective to what you may have heard about innovation. You don't have to agree with me, but I hope you get some insights into someone else's thinking, and perhaps, just perhaps, together we can change the landscape, because what I'm about to show you is not particularly good. It's actually pretty disappointing. So if I can just... Okay, so I want to start I'm going to give you 3 case studies later on, and I'm going to give you 7 issues that I've learned. But before I do that, I want to give you the background and scorecard about what this all is all about. So each year the Global Innovation Index comes out. Each year I read it and think we've improved. Each year I realise we haven't. So it's a lot of different headings, but these are the summaries they give you. So the country that we live in is ranked number 21 for knowledge creation, which is pretty good. Number 48 for knowledge impact, which is pathetic. But more importantly, for someone in my position, number 74 for knowledge diffusion is pretty embarrassing. And the amount of money, the amount of talent that we've got, we shouldn't be doing this. If you look at this next slide, what they do is a great report for nerds like me to read, great stats, but have a look at this. This is what Australia can give at the top. Number 1 for school life expectancy, number 2 for population getting tertiary training, number 6 in the world for best universities, 9 for human capital, 10 for cited papers, and 11 for publication. 23 for innovation, which is sort of okay, nothing great. 39 for granted patents, it tells you something we publish well, but then everyone read it and cite it, and then they go and protect the discoveries that are created. You don't see on the screen, we're number 78 for graduates in science and technology. Thank you, Mark, you're reading my screen. And more importantly, as I think you saw from Sally, a great opportunity of actually getting money into supporting research. We always whinge, and you'll hear later on that we don't have enough money, but we're ranked number 84 in the world for getting foreign money to support our activity. It's a disaster. You know, we speak English, we should be doing a lot more, we don't. And to me, that I'm trying to work out why, with all those attributes that we've got, and we lead the world in many areas, we just don't work. And that's what today's about. I'm going to talk about them. So why are we so poor? And a little bit about looking at some case studies, and again, some people disagree with the case study model, but looking at case studies, what differentiates success from failures and what some of the learnings. And again, this is my personal experience primarily, but I think it does translate. This is one of Australia's great success stories. Graham Clark, young child with the cochlear implant. Now, Graham took 20 years to go from end to end to get to that point. He then got cochlear involved, particularly through Paul Trainer. He took a big risk. And what have we got? We've got a great Australian company, leads the world, and there's a story about why that's successful. I'll take you through. I was heavily involved late in the piece in Bionic Vision Australia, BVA. Many of you might remember Kevin Rudd's 2020 Summit. And the one thing that came out of the 2020 Summit, they were going to give $50 million and they were going to develop the Bionic Eye. They gave him $50 million. 10 years later, there was nothing to show for it. I was brought in at the very end of that process to see what we can do with it. And I raised them another $20 million to actually get that concept into at least clinical trials, and we did it. And I'm going to take you through some of the learnings that we had from that. And more recently, I've been involved in the Bionics Institute with a collaboration with Cochlear and with Mark Cook at St. Vincent's Hospital. One of the great things that's coming out the ability to use an implanted device and continuously monitor seizures in epileptic patients could actually change the world. And you'll note, just to give you a hint, you see the patient here, he's got what looks like he's wearing a cochlear implant. It's a variation of cochlear implant, and that's something I want to come back to. So, what I've said at the beginning, I've got the Klupacs 7 principles of why we're not working as well as we should. The first one, and I learned this very, very early on in my career when I was working for AMRO John, you can have the best technology transfer people in the world, you can have the best lawyers in the world, you can have the smartest people. But what I found when I went from AMRAD to the Monash Institute of Reproduction and Development, Monash that was led by David Pretzer, it was a small department in the Faculty of Medicine. It was at New Monash. It was about 300 people of a 2,000 faculty environment. In my 2.5 years there, we spun out 7 companies. We did 3 licensing deals and we raised $30 million. Now, it wasn't me per se, but what I found was all that technology that we were able to take out in that period of time had been sitting there, and I think you were there, Margaret, was for 10 or 15 years. Right? It was there for 10 or 15 years. It just needed someone of my particular skill set to come in there, talk to science, understand it, and say, have you thought about this and this? And because no one ever had, and we've gone through it, I think you would have gone through something like that, it's putting the right people together with the talent to get that alchemy. You can have the best scientists sitting down there, talking, collaborating, clustering. It's not going to lead to translation necessarily unless you put other people in. That's something that we can't come back to. The Bionics Institute today is exactly that. We're an 80-person institute. We've spun out 3 companies in the last 3 years, 2 deals, and we've raised something like $40 million as well. Again, the reason is because the board took a view that getting another scientist in, no matter how good they were, they wanted to translate. They needed to get the skill sets of translation to mix with the engineers, give it the catalysis. They punted on me, and not me personally, but me getting other people to think outside the box has led to some of the innovations. And I mentioned before, the combination of Graham Clark, who did all that work, did all the prototyping, but if he hadn't met Paul Traynor, if Paul Traynor hadn't developed a Tiger team and nucleus, this wouldn't have gone anywhere. So Graham did a lot, brought everyone together, but it was that one person again, Paul Traynor, who did it many times, who made that happen. Culture and objectives. To me, the scientists in the room, ATSE is very, very strong about it. But you sat down and said to a car-carrying lecturer or above, why do you do science? Most of them just, I love generating knowledge. The culture of those people, when you say to them, oh, it's great technology, but let's think about translating it, they get very scared because if I do that experiment and it fails, I'm going to lose the grant. I don't know how many times I've come across that, and that culture gets in the way. The second thing that gets in the way in terms of the culture and objective, what medical research want to achieve, who do they think the customer is? To be honest, most of my medical research colleagues, their customer is fellow medical researchers. It's not patients, and that's the problem. The last thing I've got there is that Australians are brilliant analytical people. That's why we're lusted after by people around the world to become managers. But I want up in New Orleans where we've gone for the same thing. You go to America and they kick you because you're not rah-rahing, because that's not how they train, but that's what they expect. We can tell everyone why it won't work. Very rarely can our scientists tell you why it can, and that means we have a problem because we're defeating ourselves before we start. I mentioned before it's getting the right management, so some certain skills have to be in place to make these things happen. But I think the other thing that I've learned, and I've made a career now focusing very much at the front end of innovation. I've run companies that are in development, and I'm terrible at it. But the skill sets that you've got at various phases in the innovation program need to be identified if they're going to be successful. Often what happens, I don't know how many biotech companies are listed on the stock exchange at the moment, I think it's like 150. If you look at the CEOs of those organisations, all very talented people, in many cases, they've been there for 10 years. I admire their capability, I admire their stamina, but to be honest, they've all passed the use-by dates in many cases, and they need to bring in other skills to move forward. And it's that recognition. Now, some people will say Australia hasn't got enough. I disagree. The other thing that I noticed is that when, particularly in our environment of engineering and interfacing with clinicians, if you don't have a doctor, you don't have an engineer, you don't have a biologist, you don't have a mathematician, physicist, software developer, and then someone who's going to be a designer who's actually going to come and tell you how it all should fit together, you've got no chance. And yet there's so many people I come across at the university, I work with them, they're brilliant people, they're working in isolation, and then when they do the impact analysis, everyone says, well, why didn't you get the impact? Pretty clear. And yet next door to the next building is the skill set they could have had. Is bringing that together that we don't do very well. I said here, the key thing that I've learned is it sounds pretty basic. You know, when you build a house, the first thing you do, you go to an architect and say, what do you want it to look like? What are you going to do? What's the design? When we want to build a product, particularly in engineering, we know what the problem that we're trying to solve is 99 times out of 100. We can model that, we can draw it, we can do a lot of work before you even start. A lot of academic-based engineering I've come across is we'll just tinker, we'll see what comes up. It doesn't work. It's inefficient. There's still room for creativity, but that step that you would think would be so obvious is very rarely done in my experience. The fourth point I've got there is experience matters. Now, one of the things that when this concept was first looked at 20 years ago, there was a lovely report came out of QUT. It said, Australia's not very good at entrepreneurship and innovation, but, you know, as we train people in technology transfer, it will get better. It hasn't happened. And the reason it hasn't happened because unless you've got someone to learn from, it's very hard to learn on the job. Now, the best way to learn is failures, but most of our time frames for these developments take 5 or 10 years. You haven't got time to fail once you do it again. You need to bring the people in to accelerate that, and we've done a pretty poor job of bringing people back from industry into academia. And my great needs is when I go back to universities, when I go to America, I see people who've got joint appointments that are in and out the whole time. They go to Germany, they go in and out all the time. If I come here to Department of Electrical Engineering at University of Melbourne, I do not see anyone that's ever worked in industry. That tells me something, that explains it. And then you've got the concept of what they want to achieve. So at BI, it's all about, we're focused on the patient, it's about translation. University is very much about allow people to be curious. Now, there's nothing wrong with knowledge generation at all, but it's bringing that skill sets together and understanding what the purpose is. Universities, people work at universities, they don't work for. What I like to think at BI, people work for BI, not at BI. And this is our model. And this is a bit different than most medical research institutes in the country. In fact, every medical research institute in the country, because we are clinician focused. We're embedded in a hospital. We've got huge advisory boards of clinicians who interact daily with the engineers. And you're engineers here. It's amazing what happens. A 5-minute discussion, sparks happen, the next thing you know, prototypes made. We then put that around scientists, we put that around patients, we put that around a whole range of engineering skills, physicists, mathematicians, software developers, and that gives us a chance to success. It doesn't always happen, but this is a model I would love to see more of across universities in particular, rather than just the medical research institutions. Understand the missing pieces. One of the key questions that everyone comes up to, I made this great discovery, but it didn't go anywhere. And the reason is because no one ever gets 100% of what's needed for the whole product. I mean, I've got here, because I'm a patent attorney, first question I always get asked by somebody was, who are you infringing? How are you going to get the rights? Where are you going to put the money in? That's a classic. People sort of get that, but they don't do it till it's too late. But they forget, if I've made a device mechanically, if I want that to run, I need to be able to talk the various parts. You need to develop an algorithm to do X. You need to have firmware put in there. You need software to go into it. Where's it going to come from? They don't bring that in. Usability, I mentioned before, if you don't have designers, those creative people at the end, it doesn't go anywhere. You can build the thing of what you can do, the thing of engineering beauty, but the patient can't hold it. So the amount of time that should be spent monitoring the patient, seeing how it works, getting nurses in as opposed to doctors is critical, and we do that. But in terms of what's missing, very rarely it's done. The other thing at the academic level, and I know why they don't do it, but they never look at it again. Alex told me this 5 years ago. We were looking at one of the things. Go to the company, find out what they believe the gap is, ask them how much they pay, they will fill it, and then go and make it and sell it back to them for that price. Academics don't do that. And the last thing is when people are doing these things, they just assume fantastic, I've got a wonderful product, costs $20,000 to make it, people buy it. But don't forget that at some point people have got to make money. And they forget a missing part of their design is to think about, is it scalable? I make a prototype, but can I get the cost down? That's never thought about. Money. To be honest, I was struggling with it to put this in. Because everyone says there's not enough money. And excuse my my alliteration here, but I was going to say a word, but it's not true. There is a huge amount of money in this country. I just came from, and some people here just came from an audience where they're sitting in that room, there would have been billions of dollars. But that was probably one of the very first times that that group of people had been exposed to what we did. And when I left there, I thought, that was fantastic. I wish I known that. You don't always have to get a grant. You don't always have to go to government. You don't always have to beg, buy and steal at the corner store. Sometimes there are people who want to get involved and you've got to learn how to pitch to them. The real issue for me isn't money. It's how academics are taught to interact with people with money to make it accessible to them. The people in that room tonight, we went through, to give you an example, we went through the presentations we made 5 times and we just said, we've got to make it dumber, dumber, and dumber. And we felt it was the dumbest thing we ever did. I walked out and said, oh, you know, it's pretty complex. Right? But it got through. And what we think is everyone wants to know the detail. They don't want to know the detail. They want to know the big picture. Who am I curing? How much does it cost? Why are you the best? Great stuff. And we don't train our students to do that. That's the problem. And there's a huge amount of wealth now in this country that we will have to put back. The pitch that we made tonight was, look at all you guys, you're 50, 60 plus, you made money, where are your kids going to get a job? If you back us, they'll have a job. Oh, the penny drop. You know, whether I get the money is another thing, but there isn't money per se. It's how we sell it. And then once you get to that point, you know, I get this crap that comes up and says, oh, if I raise too much money, I'll own only 30% rather than 50%. And I say to them, mate, only 30% is well a hell of a lot better than 0. Right? Now, you don't want to be deluded. There are well-known venture capitalists in this country and elsewhere who will do evil things to you. But get smart people alongside you. My point is, it comes down to a team. Never walk into this. As John Parker once said, you never go into a gunfight with a knife. So when you go in to have these discussions, you pay your money, you bring your lawyer in, and when you do that, you get respect, you'll get the deal. Academics I know walk in there and think, oh, you've got to look after me. You just think people are nice. They're not. But you have to basically look after them. And the last thing in Australia, what holds back innovation, I love it in Australia. I love it. I walk around the world where we go to in the country, people respect what we do. But that democratisation of money giving is rubbish. Right? In the reality is, some people deserve to be funded, and there's some people that don't. And yet, across Australia, we all think, we'll do a little bit to him, a little bit to her, and we'd rather give 10 million to them and nothing to that person because they're not good. Oh, you'll get a bit, you'll get a bit, you'll get a bit. And then they say, where's the output? Pretty clear, that's why. Now, you can get those people together, but get them together to work together and then give them $10 million rather than cherry picking. And that's how the whole system's set up. Patience. To me, one of the things that's, we spoke about money, and Australia's got particularly investors in the stock market, long-term investors, probably 6 months. We've all been involved in programs that take 10, 20 years from concept. That's okay. We've just got to make sure people understand that along the way, you don't get to the mountaintops the goal. The journey to get there, huge amounts of innovation happen because you don't expect it, and it creates other stuff. By getting people to be patient and not expect stuff straight away. And why I raise that? Because in drug development, I heard someone talk about it just before, and John and I were involved in this many times in AMRAN. You get someone to come in, the venture capitalists will say, we need human clinical data. Give us some human data and pull back here. So they raise some money, they do a 2-3 person clinical trial, they never invest in manufacturing, there's nothing to scale, the data comes back, and they say, that's okay, we'll take it from you, and we're only going to pay you 1/10 of what you thought. And they say, why? I've got human data. They're going to have to repeat it again. Because if they've taken the money, invested another 2 years, done a 20-patient clinical trial that they could have handed to their partner, that partner would pay them that money they thought they were worth because they put on a pathway to a product. there's this thing in Australia that, doing it quickly, quick and dirty works. Quick and dirty never works, and all it does is waste time. Part of the reason our translation hasn't been as good as what it is, there's a mentality here, take a little bit and get as much as we can from a little bit, and don't ask enough. We just don't do that. I've said there, the other point I made, the second dot point, is the journey. There's a great TED talk that I saw the other day. There's been wonderful innovation made, payments and the like. There's many discoveries that were sitting on the shelf that were going nowhere until the internet came. Netflix was the example. That concept was out there for a long time, but the internet band speed was never going to be enough for the first pioneer. They failed. Internet came in. Netflix 6 months later was making bucketloads of money. Sometimes you have to be patient because you made it, your time's not right. And you just have to accept that. And you just got to be prepared to wait sometimes. This to me is a big issue, and I was really fascinated to see what some of the points that Sally made about that ability to make proof of principle and get to the proof of principle is a big issue in this country. It's not going to change overnight. I'll give you a slide in a moment about what we're trying to do with the Institute. But just because you haven't got a fully-fledged manufacturing capability, it's a problem, yes. But remember, when you're trying to partner, add value, what you just want to show is that the data you've generated has come from a consistent process, not known as a final process, and the data can be reproduced, and I've got a recipe I can follow. If you do that, you're fine. What often happens, and I've done due diligence on many people, they show me results. There's one experiment, this result, one experiment, this result, supposedly the same molecule. Why are they different? Show me your batch records. There were steps taken out of batch to batch to batch, and I said, this is rubbish. You spent years doing it together. I can't compare any of those results. Now, scientifically, you would say that's obvious. I can tell you, as a person at 35 years looking at technology, that's pretty common. Because people say, well, I made it in the bench and I didn't put this in this. I had a different unqualified batch, or the supply wasn't there. So I got the one from China this time, got the one from India that time, and wonder why the results are different. So if you can overcome it, we're trying to do something about it. We launched tonight a facility, it's a joint venture with the University of Melbourne. It's a medical device facility. We wanted to fill the gap between prototype development and first-in-human clinical device capability. It's been a gap for years. So we've built that at St. Vincent's Hospital with the University of Melbourne as our partner. We're going to expand it. It will really take time to get up, but at least that infrastructure is happening. Hopefully the government will get behind it. That doesn't get away from the fact that it's still happening. We're going to have to live with the point that I made before. Now, when I was growing up in the industry, I had a boss. I was known as Grumpy Rob, or Whingy Rob, I think it was at the time. And he used to come to my office and he'd say, I go to his office, I'm sick of this. Just give me problems. Give me problems. Give me problems. I don't want problems. I want solutions. So I'm going to try to give you my thoughts on solutions. You can disagree as much as you like. To me, there are some people who want to be entrepreneurial. There's some people who don't want to be entrepreneurial. There's some people who want to have a crack. People who want to have a crack are always going to be more successful than people who don't. In the university sector, it's a real disincentive there. If you've made an invention, you want to follow through, you want to get people around you, it's a big risk because you're not going to generate papers anymore. If you fail, the university is going to cut you. You've got nowhere to go. A US model, it doesn't always work, but a US model where if you've made an invention, say at Stanford, take a leave of absence for a couple of years, take the license with you, and if it works, great, we'll never see you again. If it doesn't, come back to us for a couple of years and we'll keep you going. That safety net, I don't think costs the system very much, and there's nothing like having a champion to give you the best chance of success. That doesn't exist in Australia, so I think it's one thing we're going to go. I'm a person who lived by these dreams. Technology transfer organisations are very, very difficult to run. Particularly the university, I don't know how many people here, but I heard that Deakin, for example, has 66,000 students and 20,000 researchers. Could you imagine trying to keep abreast of that? It's impossible. And my belief, having worked at Biopharmaceutical Institute now and run Monash Institute, one or two people very close to me besides every day of the week who've got some understanding of the industry, who can talk with them, can catalyse, things happen. But once you get past the ratio of probably 1 to 50, doesn't matter how good your technology profession is, it doesn't work. Now, I just think we can do more of it. It's really part of the TTO model, it's the centralised model in all universities. Put them out to the faculties, put them into your hubs, have a person who's there every day who understands all the competitive elements, they're hearing it every day. It's amazing how quickly people pick it up. So this is clear. We all want government to give us money. So let's be really clever about government has got a limited pool. And there's some things that we all know and the academy knows that are major. Let's band together to get government to invest in what we really need, not wish lists and people who are charismatic all the time and themselves. Let's really identify bits and pieces like we've just done. That's early phase manufacturing capability, be it drugs, be it food, be it what we're doing, medical devices. Get government to invest in that and then allow us to do it. To me, one of the great learnings of those three companies that I'll come back to now. So, Epiminder is going extremely well. Buying efficient technologies, after all the money that's been invested in, which was about $150 million all up, is still struggling now to actually get to the next stage. And the difference was bioefficient technologies never had dedicated management. There was a group of academics getting together, all running their own little fiefdoms. Epiminder, when we put it together, we did it as a joint venture with Cochlear. I convinced Cochlear to give me one of the star engineers who was middle level. They put him in to be my program manager, COO, and because he was there, everything was coordinated. He project managed, put it together. I couldn't have done it. John knows my lack of management skill. But putting him in there and putting it, I've seen in all the other subsequent companies I've been involved with, when you identify early a program manager, project manager, operations manager who's got skill and has got high EQ, these things work. When you put them, and sometimes that can be the founder, but very rarely it's not. And so to me, where is talent at the moment? That talent usually is in large companies. So what I made the pitch to the CTO of Cochlear was, you've got all these people at mid-level who are really talented. They can't grow very quickly in an organisation. You want to expand their skills. Putting to an MBA doesn't work. Put them into a startup. You can never, it's the best way to learn about skills. The startup benefits, you benefit. In that organisation, let it go gangbusters, and I think if you watch in the next 2 years, that will be on the road to being another Cochlear. For me, if you look at the grant system that we all work through, and there's all criteria on a review of the development grants, and you look at the weightings that they give you, there's very little weighting for multidisciplinary skills. I talk about commercialization. If I look at them now, I can tell who's going to be successful, or at least have a chance of success and be busted. And there's very little weighting for multidisciplinary. I think that has to be added. And I just can't, what does I say there, Mark?
Create additional incentives for impact philanthropy, angels versus pure philanthropy.
Yeah, thank you. So, like tonight, at the moment, if I donated to the Bio Institute, I get 100% tax deduction. That's great. I might even get a chair named after me. Well, I couldn't afford that. But at the moment, the incentives, and I realised most of the money I've raised in my 35 companies, the first phase came from angels or impact philanthropy. There wasn't a lot of incentive for them other than equity, and I convinced them they lost the money, at least that they made. So we've now got the E6 scheme where you go tax deductibility, but if you think about where the money is going to come from, the early stage innovation translation, it's actually wealthy people who want to have that. So let's get more of that by giving them more incentive. I'm more than happy to give someone 100% tax break if they invest in a company that fails. And I think you will find a lot of people will do it. I've also found that a lot of people do not want to donate to universities or us because they feel that we wasted money. So we can target them to be putting it alongside innovation. I think we're going to be much more successful. Getting behind the angel groups and convincing government to perhaps put dollar for dollar, I think they'll be very successful. So thanks for listening to me. I think I've gone a little bit over time, so my apologies.
Thank you, Robert. We might have some time, but we might have time for a couple of very quick questions. So, Mark, there's nothing on the chat. Is there any questions from the audience? I used to ask one question, and it's sort of a little bit off topic, but you talked about some of the models there for bringing clinicians together with engineers and scientists. And I know the Victorian government has recently put a lot of money into the ATSE Centre for Medical Discovery and Bionics Institute is a partner in that. Can we talk about when you think that approach will work or how it will differ from the Bionics Institute?
Yeah, I think to be honest though, there's two faults that we're putting a lot of money into a building.
Yes.
And the theory behind that is we're putting a building, we're all in it together, sparks are flying. That's sort of true. I think the reality is that the reason it's going to work is because the people work together for 25 minutes in a cracked out building, to be honest, the St. Vincent's Hospital, but it's worked. We'll have a nice shiny building, so maybe even better. But the reason Aikenhead will work because it's goodwill and people have worked together for a very long period of time, and people coming into that environment pick the culture up.
Yep, no, that's great. Thanks, Robert. And in the interest of time, we will finish there. So thank you all very much for your attention, particularly those on Zoom. We will be having another meeting in a month's time. I'm not quite sure of the date, but it's early June. So we'll let the Zoom people go, and then Mark, I think, has a present for our speakers. So thank you, everyone.